So it's really a great privilege to be here to be able to talk about this work. So I was fortunate to kind of grow up in science as this field was developing. As most people probably in the room have at least heard a little bit about how exciting the progress has been in regards to therapies for patients affected by spinal muscular atrophy. I'm a small part of that, but what I'm going to try to do today is tell you about the whole story, kind of from my perspective and the things that I added to the field, and kind of how that's led me to where I am now and the things that I'm currently interested in. So I do have a few disclosures. So very fortunately was involved in the ARM-STP program, and that's really probably the reason I'm able to stand here. I have also gotten funding from NIH, from the National Institute on Aging. As I'm going to talk about a little bit, I'm interested in aging-related loss of physical function. I also do some clinical work. If anybody's ever heard of the Tennessee fainting goats, patients also get that disease, myotonia congenita. And we have been looking at a therapy in patients affected with muscle hyperexcitability disorders, which I submitted a grant and was funded for that, not really relevant to this talk. And then the Neurological Research Institute at OSU has supported me. And then I have served as a consultant for neuromuscular diseases as well as spinal muscular atrophy for a major pharmaceutical company at Roche. So just about a year ago, we published our work in this area showing the first in human trial of gene therapy in spinal muscular atrophy affected infants. And this is one of the family members that was part of the trial. I have a small little story. I actually have lots of slides, so I can't tell too many stories, but I will. So this child actually was involved in the gene therapy study. I remember seeing this little girl, and the first time I tested her when she was about 6 months old, thinking, wow, this is maybe one of the first infants I've ever seen that seems somewhat cured from this disease. And I'll tell you about how the measures that I do kind of told me that. But the cool story about this is my niece actually had severe epilepsy. They live in North Carolina, but came to Columbus to have evaluation for potential epilepsy surgery. And she was undergoing the brain mapping the same time when this family was staying in the Ronald McDonald House. And my mom got to hold this little girl when she was there. So it was a really emotional, cool story that it was like full circle. My mom got to meet this family, and they were just talking, and my mom was like, oh, my son's involved in that trial. And then they found out I was the common denominator there. So really, really amazing thing to be part of. So what I'm going to try to do is give you an overview of spinal muscular atrophy. I imagine a lot of people in the room know a good bit about this disease, but I'm going to give you kind of the genetic underpinnings and the mechanisms as far as we know on why low levels of survival motor neuron protein cause muscle or motor neuron disease. And then I'm going to tell you about kind of the progress in the field and how it kind of led me to becoming interested in loss of physical function and sarcopenia of aging. So spinal muscular atrophy is, generally it affects infants, the most common form. So it results from motor neuron death, and so that causes muscle weakness and atrophy. It affects about 1 in 10,000 live births. It is an autosomal recessive disorder, and patients with more mild symptoms can often have this fine tremor, which seems very stereotypical between patients. The genetic basis of spinal muscular atrophy is an homozygous loss of the survival motor neuron 1 gene. So this is usually a large deletion of exon 7 and 8. But in some cases, you can also have a small missense mutation. But generally, genetic testing for large deletions will pick up 95% of SMA cases related to this gene. The really interesting thing about this disease is there's actually 2 genes involved with this disease. And patients with SMA have homozygous loss of SMN1, but a backup gene, SMN2, and we'll talk about how it does this, modifies the disease. And so the number of copies of SMN2 gene modulates how severe the disease is. So there's inverse correlation. More copies, less severe disease phenotype. This is a mapping of how most people characterize disease severity, and I would throw the big caveat here. This was pre-therapeutic era. So type 0 is relatively or extremely rare, has prenatal onset, and infants affected have need for respiratory support at birth. The most common form, about 50 to 60% of SMA, is type 1 that has onset in my experience, generally 6 to 8 to 12 weeks, but it's basically onset before 6 months. These infants never achieve ability to sit independently, and their natural history of death without invasive support is usually less than 2 years of age. The median survival is closer to about 8 to 10 months without support. And then from there you go to less and less severe phenotypes. The one that I've actually become most interested in, and our center is one of the leaders investigating this, is type 4, type 3B or 4, and that has onset during adulthood. So a very, very mild phenotype. Represents maybe 5 to 10% of all SMA, so it's relatively rare. This is a phenotype that can be relatively challenging to diagnose. It can mimic things like other motor neuron diseases such as ALS, and so this is one of the areas that we've really been particularly focused on. So I've told you that loss of SMN is present homozygously in patients with SMA. So when you look at the two genes of related to SMA, you have SMN1, so that's on both alleles, that's lost, usually a large deletion, sometimes a small missense mutation. But then all patients, there's never been a patient reported ever with SMA or otherwise that has loss of both of these genes. So it's incompatible with life, seemingly, to not have some version of SMN2. But when you have SMN2 only, SMN is lost. The problem is this gene is kind of what I call a wimpy backup version of SMN1. So it's in, essentially, there's a single nucleotide change in exon 7 that causes the majority of the transcripts to only produce this short truncated version of the protein. About 10 to 15% of SMN2 is translated into a full-length SMN protein, but the bulk of it, 90% or so, is this shorted, truncated version that rapidly degrades. And so basically produces small levels of functional SMN protein, where this produces predominantly 100%, but in SMA patients, they don't have this. So when you look across phenotypes of SMA from very severe to more mild, generally this type 0 phenotype is associated with only one copy. So you may ask, how does one copy happen? Well, so some patients have no copies of SMN2, but they have SMN1, and that is essentially that's associated with the normal phenotype. So one parent basically had one copy of SMN1 and a large probably deletion of SMN1 and then no copies of SMN2. That's still compatible with normal function, at least as far as the superficial level. But then the other patient may have had, or other parent may have had one copy of SMN2 and then the homozygous deletion of SMN1. That's pretty unusual. The most common allele frequency in the population is one allele of SMN, or one copy of SMN2 on each allele. But this region of the genome is very unstable, so there's a lot of duplication. And so you have some patients that have no SMN1, but they have a very high copy number of SMN2 and have extremely mild phenotype, maybe even normal. And so this genetically has been shown to basically rescue the phenotype based on this backup wimpy version of this SMN gene. So how did I kind of get interested in spinal muscular atrophy? So I, as similar to Tay, so Tay and I have worked together for the last several years. He's doing great work in aging, and we have that in common. But we both did neuromuscular training after residency. And so after residency in 2008, I did a neuromuscular fellowship at The Ohio State University and then joined the faculty thereafter. So I was really primarily a clinician between 2009 and 2012 or so. I was really in the clinic full-time, seeing patients, but I was curious about a lot of different things. And I had been kind of rubbing shoulders with physician scientists and scientists who were doing work in this field. They had developed an animal model, which I'll tell you about, and there was a therapy that was looking really promising in mice. And so what I kind of started wondering, is there ways that I could use my clinical skill set to kind of take from the bedside to the bench to help these scientists ask very clinically translatable questions? Are there quote, biomarkers? So biomarkers are, it's a buzzword, we hear it all the time, but really it continues to be important even after you have therapies to understand the disease. So they help us with diagnosis, so that's SMN1 homozygous deletion, that's SMA. Prognosis, that could be SMN2 copy number, so it gives you a prognosis on how severe the phenotype would be. And then it's possible that you may have markers that can tell you the disease state and like how likely is it that you would have a response to a treatment. and that may help you stratify to different types of treatment. So in this case, associated with low levels of SMN, but maybe there's other pathways that we could augment at different stages of the disease. And then these measures can also quantify a response and help us, modulate our treatment accordingly. We use that in autoimmune neuromuscular diseases all the time. And then my main goal is to help understand the biology of SMA. So right now, there are several different types of biomarkers. So we have molecular biomarkers, which I mentioned. We have measures of muscle mass, which are simple. The one caveat there is muscle mass doesn't always equal muscle function. Sometimes muscle mass doesn't really correlate with strength that well because you can have reduced muscle quality. But the main thing that I thought and wondered about is, like, can we do some of the same measures that we do in babies with SMA in mice? Because in mice, we have genetic tools that we can manipulate pathways. We could track them basically through the whole phase of the disease, pre-symptomatic to post-symptomatic, and then do interventions to see what kind of effects we could have. So what I thought is, but maybe I can create a little mini EMG mouse lab and test these mice and see what happens. And so that was kind of very simple on the surface, but what was surprising is nobody had done it. So it wasn't anything earth-shattering, but it was kind of like my doorway into getting into science. And from there, I've tried to push myself farther and farther, but that was kind of what made me curious, can I do this? So electrophysiologically, we have a lot of measures that we do in the clinic. I do those. I do a lot of EMG every week. We take these for granted. There's actually a lot of data that I think in the clinic that we don't even leverage. So we have simple things for instance, CMAP, compound muscle action potential. You stimulate a nerve, you measure it from a muscle. It's basically a summation of the excitability of muscle fibers. And normally we look at the speed of the response, so how fast the conductivity along the nerve takes from stimulation to recording. And then we simply look at just the size, basically the output. And that tells us a lot, but there's a lot of other parameters that are basically untapped that may tell us more things about the disease. But basically what I did is looked at these measures and tried to understand, for instance, CMAP, functional status of the neuromuscular system to cause excitability of the muscle fibers, so connectivity at the NMJ, numbers of motor neurons, number of axons. Because this is kind of an indirect measure, because of collateral sprouting, the CMAP can be normal, but you can have reduced innervation. So motor unit number estimation is an estimate technique that allows us to get insight into the number of motor neurons that are functionally connected to the muscle that you're interested in. Very cool, because there's not many things in biology that you can actually track a population of cells in vivo longitudinally. And we could apply, and so basically I was interested in applying this to animal models in the way that we had done in the clinic. Now, of course, it's an estimate, but that's very powerful because motor neurons, as you know, are non-dividing cells. So the motor neurons that we're born with are basically there for our lifespan. So when they degenerate, there's no known pathway for regenerating those, so you can really get a clear understanding of the connectivity in the neuromuscular system longitudinally. The rationale that I had for kind of investigating these measures is we knew from clinical studies that when you looked at compound muscle action potential, simple, just ulnar stimulation, the hand muscle here, it correlated very well with motor scales that we use in the clinic that are quite complicated and involved. So this is a simple measure, totally objective, that could give us insight into the status of the neuromuscular system. We completed around the time when I was starting to get really interested in mice. We had a multi-center study. A collaborator, his lab is just down the hall from mine, Stephen Cole. He's MD, PhD, kind of my inspiration to try to kind of learn how to be like him. So he was not very senior compared to me, but he was kind of like a parallel peer that mentored me. And so he designed a multi-center study where we did these measures in SMA babies longitudinally. And you can see that when you compare healthy infants with normal SMN gene and then infants that have 3 or more copies versus 2 copies, you can see a very striking pattern of motor neuron degeneration there. I think the biggest motivation to taking this leap, though, was the fact that in 1995 we discovered what gene caused this disease. In 2010, at OSU and Nationwide Children's in Columbus, Ohio, the first dramatically successful therapy for SMA in a mouse model was published. So this was an adeno-associated virus, subtype 9, to basically deliver cargo of the SMN gene to motor neurons and rescue the phenotype. And this took a mouse that normally lives about, on average, 13 days to living over 500 days. And so this was published a couple years before I got interested, but this was really catching momentum. And so I wanted to investigate these therapies using a in vivo model and measures that kind of paralleled what we do in the clinic. There was this concept at the time that SMA is a very unusual natural history kind of progression disease. So most patients, by the time they're diagnosed, have striking neuromuscular defects. You can see the previous graphs I showed, the longitudinal. Those were infants that were diagnosed and then enrolled in a clinical trial. They were already dramatically separated out. But they're unfortunately only a handful, but there are a handful of infants that have been studied pre-symptomatically and then tracked over time. I've seen a couple patients, 3 or 4 patients like this, and this was in the literature at the time where basically these 4 infants had been studied kind of early diagnosis because of a sibling that was also diagnosed with SMA, so they were identified before disease onset, and they tracked them over time. So this kind of colored area here was the question is there this pre-symptomatic era, time period when possibly we might have more impact of disease modulation. So the mouse study that had come out in 2010 clearly showed that with the later time point, the mice didn't do as well. The big caveat there, though, is the virus in mice targets more glial cells than motor neurons after day 5 after birth. And so there was this question of early versus late therapy, which I was very interested in. So one thing that's always good when you're getting into an area is try to link into a mentor that can help you kind of delve into the field, learn the background, and then quickly you can kind of become an expert by synthesizing the data that's already out there. So my primary mentor, he's a PhD, he's a British, Arthur Burgess. He got invited to do a review, and so he asked me to be involved with this. And so I basically dug into this area. I had been getting more and more interested in it, and it tried to synthesize this idea of pre-symptomatic versus symptomatic versus late symptomatic treatment. And what we kind of tried to character or kind of hypothesize regarding was these different phases of disease, how much effect could we have on the disease phenotype. And so we predicted with early treatment, maybe if there is this pre-symptomatic window, can we get full effect? And then as you go later and later disease, basically the horse is out of the barn and you lose more motor neurons. Motor neurons are not replaceable, and so you may have less and less effect. And so this is kind of what we predicted as I was designing my career development award to try to test these concepts in a mouse using clinically relevant measures. So this is kind of my bedside to bench to bedside approach. So on the right here you see a picture of our EMG lab, and then on the left and then right here you see a picture of my lab that I have kind of like a little mini mouse EMG lab. We do physiology and various other measures. So over the course of the last 3 or 4 years, we've taken this from humans to mice to neonatal mice. We have a PIC model, which I'll talk about. And then this is one of the clinical trial infants that you can see that he's standing up. So I'll show you a video about that later now. And this is a type 1 baby that should not live to his 2nd birthday. So our first paper was basically just trying to define the natural history of the disease in mice. And so we showed that actually early on between day of birth, so we started measuring at P3, so I had to really miniaturize these measures. So between P3, 3rd day of life, to P6, the measures corresponded pretty well with wild type, and then you see this divergent. And now this was untreated, and then we did early therapy with adeno-associated virus to deliver the SMN1 gene, and then we also used an antisense technology, which I'll talk about, to restore SMN protein. We showed that when you treat early, you abolish the disease phenotype in these mice. And this was the first paper that I kind of leveraged these techniques to understand SMA. So from there, we wanted to understand early, delayed, and then late treatment to kind of see how that affects between the different phenotypes. So this was using what we call the Delta 7 mouse model. So this is a mouse that actually was created in 2005 by Arthur Burgess, who was my mentor. So he was really familiar with this mouse and the phenotype that it had. And so we had previously described that the electrophysiological phenotype started at P6. And so our goal was to kind of go right after birth, 0 to 2 days, P4, and then P6, kind of see when it's really obviously abnormal. We use antisense oligonucleotide strategy for this approach. So this is basically a nucleotide sequence that binds to an area around the gene that makes the SMN2 gene act more like SMN1. So instead of only producing 10 to 15% of the full-length protein, it makes it produce more like 100% of the full-length protein, so increasing SMN levels using this antisense strategy. So when we did this experiment, we tracked survival, and you can see that the diminishing returns the later you treat. We also weight as a good readout for these mice, and you can see that the same kind of strategy there. And then we did electrophysiological connectivity, and you have CMAP and Muni. You see that with CMAP, it's reduced, but it's almost starting to normalize because of collateral reinnervation. But with delay, you can see there's loss of motor unit number with the later treatment. So this suggested that early treatment is probably really, really critical. Exactly how this aligns with the natural history in humans may vary between individuals depending on their copy number, depending on other genetic factors. So we know there are modifiers in the region and outside the region of SMA gene, so different patients act different. So just because they have 2 copies of SMN2, it doesn't mean they're always going to act like the classic textbook SMN1, but generally that is a pretty good association. So after the mouse model gave us good data, we actually used a different strategy. So there was a group that was trying to generate a pig model of SMA. So I'm not going to go into this detail too much, but humans are the only vertebrate species with 2 SMN genes. So mouse model had to have a transgene from human put in with SMN2. Pig model, we were hoping to have an SMN2 containing pig, but because of a series of complications, that didn't happen. So the strategy that we took instead is we used adeno-associated virus. So that's a non-integrating viral vector to deliver short hairpin RNA to knock down pig SMN protein. Then we did early and late therapy with adeno-associated virus to put the human SMN back in. So this short hairpin RNA is specific to pig, and then we put the human in to replace it. So we did early and late. So we injected at P5, so you can see this picture here. So I'm not an injection type person. They made me do it in residency. I didn't love it. But I learned how to do pig cisterna magna injections, and it was actually really cool to see how it outlines the CSF space. So this was a CSF delivery. This virus has specific or relatively good tropism to motor neurons, so it's really good at sending its cargo to motor neurons, and that's the power of this strategy. So we treated early and late. Without treatment, we had a really nice phenotype, so we did CMAP and Muni, so we translated that from human to mouse to pig, and you can see really striking reduction with knockdown, and then pre-symptomatic treatment, you can see it almost normalizes those measures, really good, real simple measure. And then we also did morphology showing ventral root loss and motor neuron loss. I'm going to show you some videos if I can make these work here. So this is basically a ramp, and the pigs go for like sweetened condensed milk. It's at the top, so they really do like to run to the top here. And you can see that's a normal looking pig, and you can see early rescue here. Looks pretty normal. This was, now that I've seen these therapies in humans, this used to be super exciting. Now it's like, okay, great, let's get to the human stuff. And then here's late rescue and no rescue, and you'll see that the late rescue looks pretty good, but definitely has some defects, which I'll show you electrophysiologically here in a minute. So you can see some hind limb kind of weakness and slipping there, but overall pretty good. And then no rescue, you'll see, unfortunately cannot get up the ramp really whatsoever. So the weakness started in the hind limb with this phenotype and progresses to the forward limb. When we do electrophysiological measures across control, knockdown, early treatment, late treatment, you can see there is this kind of diminishing return on the number of axons on ventral root counts. And electrophysiologically, I thought it was really cool to see how these really matched up with each other. So Muni and anatomical counts, there's really no good way to validate each of those, and so it's more conceptual in a lot of ways. But these really gave us a lot of confidence that these might serve as a really good biomarker in clinical trials. So basically what we showed is there's a clear electrophysiological phenotype in models of SMA, pig, and in mouse. When we treat early, we have larger effect. When it's treated late, we have less effect. And if it's very late, we have no effect. And so the the uncertainty of what would happen when we took it to the clinic was really exciting to me, because I was really interested to see how this matched up with the first-in-human trials. So I wasn't involved in the antisense oligonucleotide therapies, the nusinersen. I'm going to present some of that. But I've been involved primarily in the gene therapy strategies. Gene therapy is earlier days, but it's currently under determination whether it's going to be FDA approved. Nusinersen, the ASO therapy, is currently FDA approved as of a couple years ago. So I'm going to kind of talk about the time dependency of response in the early clinical trials of both of these therapies. So when you look at this, it's kind of a busy slide, but what this is comparing is the different clinical trials of nusinersen. So Spinraza is the label name. This is FDA approved for spinal muscular atrophy, all types. But you can see the clear differences. So this is basically a motor scale, and higher is better. And this is the pre-symptomatic therapy versus the symptomatic clinical trials. And so clear difference between those. The question is how close to normal this is getting. Probably not normal. And so the way they define pre-symptomatic, they use basically compound muscle action potential, which I've been doing animal and clinically, as a cutoff. And they said 1 millivolt or higher met criteria. In my experience, though, 1 millivolt is not normal. So most of the time when I've studied pre-symptomatic infants, their CMAPs have been in the range of 5 and 6. So their definition of pre-symptomatic may not have been fully pre-symptomatic. It may have been early symptomatic. So we really don't know, like, early as possible, so maybe shortly after birth, how sufficient is therapy going to be. But in these cases, you can see dramatic improvement with early and late. I think the coolest thing when this went to the clinic is actually, I think, later treatment in humans seems to have more effect than the mice did. So normally we see this loss in translation from mouse to human. But in this case, I actually was really, really surprised that with these late treatments, we still got response. I thought that was really amazing. So one is clinical diagnosed SMA, the other is like genetically diagnosed. And it really depends on like functional status. And the pre and post-symptomatic was really only defined in this trial, and they used the compound muscle action potential threshold. And they used 6 weeks of age. So 6 weeks of age and CMAP amplitude as kind of their threshold. So less than 6 weeks and CMAP greater than 1 millivolt was their criteria for pre-symptomatic. And the other trials were, they just couldn't be like event dependent. So basically it was any stage of the disease. So some of these infants were quite late, 6 months of age, 8 months of age, and so on. Does that make sense? Yeah. The same kind of pattern that we saw in nusinersen, we also saw in gene therapy, where you see this is the low dose cohort. So this was a first in human phase 1 FDA trial. It was really cool to be involved with this. I saw these infants at baseline, 1 month post, and every 3 months. So all these visits, I got to kind of see this progress in the field, and just had to keep it to myself, which was really exciting. But you can see clearly that there's different ages of when these infants were enrolled, and it shows a striking association with age and response. And there's a paper that's hopefully coming out soon that's going to detail some of these changes more, because just because these were younger didn't always mean that they were more functional. So it didn't even always line up with how functional they were at baseline. It would seem to be really closely associated with age. Now, the one thing I would say is these two infants were clearly the ones that were least symptomatic when they were treated, and you'll see some videos of one of these infants in a minute. But yes, yeah. So CHOP and 10 is just like a motor function scale, and so lower is bad, high is good. Generally, no type 1 SMA infant ever gets above this 40. So the fact that they're shooting over that and then they're hitting the ceiling here is really just amazing. I mean, it's really, really incredible. Yeah, good question. Yeah. So in this, we actually, all the data suggests that it's going to persist, but we don't know. So in this strategy, you're using adeno-associated virus to target motor neurons. We've done a lot of work in mice showing that SMN levels really, if you increase in motor neurons, that's all you probably need. We've done a series of Cre-Lox-P experiments, like deleting and increasing in different cell tissues and showing that a few years ago. But motor neurons are non-dividing cells. It's a non-integrating vector. So in dividing cells, you're going to lose it. In non-dividing cells, it should persist. Could it become inactivated down the road? It's possible. We just simply don't know. But in theory, this could be a one-time treatment to restore SMN levels in motor neurons. It's more about putting the gene back in. So it's a self-complementary like setup, and it's under a continuous promoter. So it's basically the AAV delivers the cargo of the gene to the cell, and then it's there. So that cell is producing its own SMN. Now in dividing cells, it's a non-integrating vector, so it doesn't go into the genome because there's concerns with that. You could hit an oncogene or something like that. But this, in motor neurons, because they're non-dividing, it's just sitting there, and it should, unless it becomes inactivated, which we don't know, it should produce SMN protein lifespan, as long as the cell is alive. Yeah. Maybe. Yeah, maybe you need more requirements, or who knows. So this is the girl that my mom got to hold when she was much littler than this. And it's not, sorry about the video quality. This was the parents that took it. But she, you know, it was, well, I thought I clipped that. Sorry about that. Okay. So you can see that she has weakness. I think anybody who deals with pediatrics can see that she's clearly weak. But she was treated definitely after symptom onset. And to me, that's amazing that you get this kind of rescue. So I would say she may eventually be phenotypically more similar to like an SMA type 3. So partial motor neuron loss. But compared to type 1, that's amazing. Now, then this other child is 14 months here. I can tell you a few months after this, he was run away with from me when I was trying to do my measures. So this is like right when he first started walking. So now he runs and he can reach up and touch the elevator. I mean, it's just like he looks normal. She looks dramatically improved. And now, did all patients get this much improvement? No. And this is because these are the 2 youngest patients that were treated. So there's a paper, as I mentioned, coming out that will give more details on these cases. So crazy exciting progress in the field. I was like really fortunate to be able to grow up in science as this was happening. It taught me how to be critical, to work in the lab, to build a team. And then I also got to see miracles like this, which were really triumphant in clinical trials. The question is how sufficient these therapies are going to be, and I think we still don't know that. So the one thing that I would, so people probably in the room are familiar with polio and post-polio. So if you partially lose your motor neuron pool, the other motor neurons can compensate and collateral reinnervate. So on the surface, you get the tip of the iceberg, and you may look kind of normal from a functional standpoint, but as we age, you lose motor neurons, and so it's possible that they could have accelerated aging, and they could have like a sarcopenia phenotype. And that's what probably causes patients with SMA to get worse as they get older, at least part of it. We don't know that for sure. So we still need good measures of motor function to track that, because what if their AAV quits working, but we don't know it, and then their SMN levels are dropping, and then all of a sudden, like if anybody's familiar with ALS, you don't really get clinical defects in muscle until you have striking loss of motor neurons. So you could just have this SMN levels be dropping, and the motor neurons could be dying, but collateral reinnervation is kind of keeping up with it, and all of a sudden, devastated. So we still need better markers and better understanding of what happens in these patients. We need to know SMN levels clearly change during age. In young development, it's really high, and then it goes down. Seems to be important for neuromuscular junction maintenance and repair, but is there a way to track how much a patient needs? And so that's really challenging. So people are working on that. And then, as I mentioned, could we have an accelerated aging phenotype in the girl that was partially treated but not completely normalized? Shifting gears here a little bit, because SMA kind of sparked some interest in the role of motor neuron function as we age. So there's a lot of clinical similarities between spinal muscular atrophy patients and age-related muscle weakness and atrophy. And because the motor neuron is a non-dividing cell, there's no way to replace it. So that has to do a remarkable task. So if you think about the size of a motor neuron, it's 1/10,000th of a meter. And then that single motor neuron living in my spinal cord sends an axon all the way to the muscles in my foot. it has to maintain that connection for my whole life. In addition to that, motor neurons during contractions, depending on what type of contractions you're doing, for a ballistic contraction, it may fire up to 100 times a second to cause the muscle contract. So very energetically active, maintaining the NMJ a meter away, and having to do that for my whole lifespan. So I was really interested, what happens to the motor neuron as we get older? And so there are some studies that have looked at this, and I'm going to kind of walk through some preliminary data about some of this that we've looked at, mainly in mouse models, but a little bit of clinical data that other people have also established. So sarcopenia, how does it affect individuals and society? So sarcopenia initially, which is described as loss of muscle mass, has become more equivalent to muscle mass and weakness because people have shown clearly that muscle weakness during aging drops faster and that muscle strength is more tightly associated with outcomes. So if you look at mass, the amount of explanation that mass provides for people related to outcomes and functionality is not very good compared to muscle strength. So basically, muscle mass doesn't always equal strength. Sarcopenia has a dramatic impact on individuals, large increase in impairment and loss of mobility, but also associated with all-cause mortality as well as cancer mortality. And the cost to the U.S. is significant. So prior work, about the time when I was born, I was looking at motor neurons and how those change across the lifespan. And so, as I mentioned, non-dividing cell, no way that we know of to replace those. And there is this capacity of motor neurons to reinnervate and compensate. So each motor neuron actually has about 2 to 3 times the ability to reconnect the muscle fibers. So if a muscle has 200 motor neurons, you can lose up to about 2/3 of those and still maintain pretty well your function. So kind of that's related to the post-polio phenomenon. But during aging, there's known to be loss of motor neurons and motor units and impaired reinnervation through a series of experiments. So 2 groups, as I mentioned, showed that across the lifespan, there's significant loss of motor neurons. They showed approximately 25% when including all individuals. But clearly, there was variability between individuals. So some individuals had greater than 50% losses. This is important to know because, and actually, there's not a lot of clinical data out there. Patients that have sarcopenia, not everybody, as they get older, gets, quote, like diagnostic meeting criteria of sarcopenia. And most of those criteria are based on cut points of populations. But some do. And in this study, they showed that some individuals seem to have more propensity to motor neuron loss. So is that genetic factors? Is that environmental factors? Is that inactivity? What's driving that? And so the big question I have is a sarcopenic patient versus a non-sarcopenic patient. How's their motor neuron count, motor unit count line up with that? Subsequent other groups showed that when you look at motor neuron counts and ventral motor axon counts, also showed reduction in aging. Electrophysiologically, one of the first studies after motor unit number estimation was kind of described by McComas in Canada. They looked at it across the lifespan, and they showed clear loss in older adults. And so this was kind of the rationale for looking in mice in my study. Single fiber, which Tay and I have both been working in great deal with, looking at neuromuscular junction transmission. So in the clinic, I do a lot of single fiber in our EMG lab. I'm really interested in neuromuscular junction disorders. And this has been shown to have defects in aging. This hasn't really been looked in really detail, and Tay's actually working on that right now. One study using a different technique just recently reported that when you look at sarcopenic individuals versus non-sarcopenic individuals, it seems that sarcopenic individuals seem to have more impairment of neuromuscular junction reinnervation, which I found interesting because that suggests that maybe you lose motor units and you can compensate in some individuals, but if you can't compensate, then maybe that's why some people have more susceptibility to sarcopenia. So all that as the backdrop, my hypothesis kind of moving into the aging world was whether or not loss of connectivity of motor neurons and the motor unit could lead to loss of physical function with aging. And really didn't have any hypothesis where that's coming from because the problem with the motor unit, you have so many different factors that are interacting. And a lot of people in these fields are either muscle people or motor neuron people. So what I tried to do is have a systems-based approach to look at the whole motor unit and try to figure out, is it coming from the muscle, synapse, motor neuron, or all of the above? So our approach, so back in about 2013, 2014, when I was working with SMA, the biggest key with a career development award is to always be thinking about the next step. Because a career development award, depending on who you are, that might be your first step towards independence. For me, it was learning how to be a scientist, and my independence step was going to be next. So I was trying to think of the next concept that my lab would be investigating primarily. So really, investigating motor unit connectivity was that for me. So back in 2013, 2014, basically got 10 old mice, as old as I could get. It's around 10 months, which is actually not very old, and just started tracking them longitudinally to see when their motor unit connectivity degenerated and how that was associated with muscle mass and muscle contractility across the lifespan. So using these measures, small numbers, but we showed, and these are repeat measures in the same mice, so we tracked them about every couple months, and we showed that actually loss of motor unit connectivity was a relatively early phenomenon, and so we recently published this work. And so a couple interesting findings with this. So 20 months of age is relatively young for a mouse. The single motor unit potential, the SMUP, is the size of a single motor unit, and I expected that would go up with the reinnervation during aging, but that wasn't a clear signal. So it made me wonder, is there a loss of reinnervation capacity in aging mice? But clearly the motor unit number reduced. We also looked at in vivo imaging with MRI. So I got a small pilot grant to do MRI of the hind limb, and we quantified the muscle. I had an undergraduate student that learned how to do this, and in small numbers, but it was very interesting that we saw a very striking association between muscle volume and motor unit number in old mice. And then we next wanted to look at, so sure, it's great, motor unit number estimation is a nice electrophysiological measure, but we wanted to kind of branch into actually muscle function. So in the last decade or so, there's a technique that allows in vivo assessment of muscle contractility. You can do this repetitively over time. So kind of like the final common pathway, what we really care about is how is the muscle functioning. So we want to look at muscle function compared to motor unit number. And we investigated this. This study was for simplicity and efficiency. This wasn't a longitudinal study, so we took cohorts. We had previously shown that motor unit connectivity started at 20 months, so we wanted to take kind of midlife, early, mid, and kind of early aging to kind of see how these measures were associated. So this measure is, in essence, it's ridiculously simple. So it's like a little accelerated pedal that the mouse's foot kind of is taped to, and then you stimulate the tibial branch of the sciatic nerve to get plantar flexion, or you can do the fibular nerve to get dorsiflexion. And you can measure twitch torque or tetanic torque. You can see here repetitive twitch responses at 25 hertz. As you increase the rate, you get a summation of the twitch responses to get a muscle output. And so this kind of gives us assessment of muscle contractility, muscle strength, as well as force frequency of relationships and things like that, which would give you insight into muscle fiber type and things like that. And so when we did this measurement, we showed at the age where we see the motor unit loss, we do already see motor unit drop. So you can see that that is reduced over time. Motor unit number is reduced. We looked at the association between motor unit number and muscle contractility, seemed to be pretty well correlated. The one big caveat, and I think this is, I admit this is a weakness of this is all the mice pulled together, so there's multiple variables that are changing between the mice. One is age, one is the motor unit number and the contractility. So this is a bivariate correlation, so you have to consider this could be primarily driven by age rather than the interaction between the two. But still interesting, doesn't tell us cause or effect, but still gives us some exciting information to kind of go on. So muscle size and muscle contractility seem to be connected to motor unit number. So the question is that maybe a target that we can kind of address? And so that's basically the approach that we've been taking. In the last few minutes, I'm going to kind of tell you about a very serendipitous situation where kind of both of my worlds kind of came back together. So it was kind of a full circle. So in 2014, there was a group. So Umrao Monani, he's at Columbia. He actually was a postdoc fellow with Arthur Burgess, my primary mentor, developed these mice that you could turn SMN levels up and down at different time points. And what he showed, if SMN levels are low through development, they develop SMA. If you allow them to develop for the first 3 weeks with relatively high levels of SMN, they look normal. And then if you turn it down, they develop this kind of early phenotype of aging. They also did experiments where they did peripheral nerve injury and showed that the mice with low levels of SMN seem to have impaired nerve regeneration. So these studies suggested that there's an early period when SMN levels are really needed. This might be relevant to SMA. It also showed that SMN levels seem to be important for neuromuscular junction, like maintenance as well as repair. And so I'm a big runner, endurance athlete kind of person. So I was doing a long run out in the woods. It was really bad weather, so I probably shouldn't have been driving. It was super icy. And my friends that were going to run with me didn't show up. But I remember right when this thought hit me, I was like, well, we have mice down in the vivarium that have high levels of SMN. How do they age, and how do their nerves regenerate? So basically, I went right back, started thinking about the experiments. I basically transferred them to another protocol that had nerve injury in it and went straight away to do these experiments. Super excited. So let me tell you a little bit about these mice that we call high copy or high C mice. So in 2000, so almost 20 years ago, these mice were generated where there's low levels of SMN, and then they have mice that actually have 8 to 16 copies of SMN2, so that they have very high levels of SMN protein from this backup gene. And basically, these are the mice that were down the vivarium that I was interested in. When I started digging, actually, this is a scan of a census and lifespan from, I think, 2003, 2004. I looked at the average lifespan of these mice, and when you compare this, take it with a grain of salt. When you compare this to prior lifespan studies, these mice are on the FEV background, so those are like white mice. Their average median lifespan was between 25 to 50% longer than wild-type mice. So, when you overexpress a protein, you're always worried about a toxic effect. I wouldn't say we can argue too strongly that this increases survival, but at least it's not a negative. But maybe it's a positive. They also seem to have generally larger litters, and they seem to be bigger. And that's the opposite of aging. So aging mice, the Methuselah mouse, is tiny. It's a little dwarf mouse. And so this kind of breaks the rules in aging, which is, to me, very interesting. So I did a couple or 3 different experiments to kind of dig into this, and this is all early days. So what I just kind of want to tell you the next step of my journey towards becoming a physician scientist. So the first experiment, this was the experiment I did in late 2014, early 2015. I believe that's when it was, showing basically peripheral nerve injury. So this is a nerve injury of the sciatic nerve, and then we track these measures in vivo. So this is CMAP immunity. At this point, we didn't have muscle contractility, so I couldn't do those measures. So how this aligns to muscle contractility, it's hard to say, but it was actually really striking how much of effect. We have been doing nerve injury in other paradigms, and so we had this kind of optimized, and so I quickly applied this to this question, and it was really quite striking. So right now, we are generating mice so this is transgenic. They always express high levels. What we want to do is have mice that we induce SMN levels after nerve injury, so it's more translational. Next thing, small numbers. Again, I apologize I don't know why my graphics have gotten distorted here, but basically we have 4-month, 18-month, and 26-month mice, and you can see that CMAP immunity and aging seem to be improved. Immunity more than CMAP. I don't know what to make of that, but that was pretty encouraging for the aging responses. This is just showing you the graph of the survival. And then the last experiment, which I'm actually most excited about, but it's also the most challenging. So basically, we have generated mice that have about wild-type level SMN protein using a combination of knockout and transgenic alleles. So we have done ECL ELISA assays for SMN levels in different tissues and shown that these are around 1 to 2 times wild-type. So they're a little high at baseline, but we're taking these, aging them out for 26 months, and then treating them with a small compound, RG7800, which has been developed for inducing SMN levels in SMA patients, and treating these mice after they're at a relevant age, or 26 months, which is really old for a mouse. This is just a schematic of how the drug works and increases full-length expression. And this is the drug molecule. This was generated by Roche, but we actually had it generate. We had somebody compound this for us, and so this is parallel to Roche what they're doing. And then we have done these preliminary experiments. So electrophysiology, small numbers, can't get excited about it. Really no difference there whatsoever, but the red is treated, blue's untreated. But then what was really cool is the muscle contractility, and these were all done blinded to treatment. We did vehicle and RG7800 using oral gavage. But muscle contractility seems to show a pretty striking effect. So it's not surprising that motor unit number wouldn't be changed because that shouldn't regenerate. But NMJ connectivity might improve. And so when you normalize this, I didn't show this, but when you normalize this body mass and to muscle, it shows the same signal. So what that suggests is maybe improved muscle quality, so improved contractility because maybe the NMJ connectivity. We also need to look at the muscle effects directly as well. So I'm going to kind of come to a conclusion here. And so I think I've been able to show you the excitement in the field of SMA and how much of a privilege it is to be able to do this in mice and pigs and then humans, and how it's really a great paradigm that I think can be reapplied to other diseases. I would kind of caution that SMA in a lot of ways is primed for success because you have this built-in target, but I think it's been amazing to be part of that. And then I think there's still a lot of things we need to do to understand SMN across the lifespan. Whether or not it improves connectivity with nerve injury and aging, that's a different story, but I think for SMA this is also important. And so what this would tell us, I think, is it may be that we don't yet know the optimal level of SMN protein. So for SMA patients, we need to know how high we need to take it before we're going to get the optimal effect, and when that needs to be high. Does it need to stay high through lifespan? Those are unknown questions, but I think this work will hopefully get us closer to some answers in that regard. And really the main summary of my lab is it seems that motor unit connectivity is important for aging. So that's like a target that we're using to try to develop therapeutics. I also wonder, could sarcopenia be a motor unit penia? And so could MUNI be a good biomarker in aging studies? Because everybody's focused on muscle mass, but maybe we need better mechanistic related biomarkers. And so we're hoping to move this forward. So right now we have some experiments where we're trying to look at SMN levels across the lifespan in motor neurons using laser capture microdissection. So basically motor neuron slices where you use a laser to get tissue from just motor neurons and kind of enrich sample and see if that changes across the lifespan, because we know that it changes during development, but we want to know in old mice, is it low? And that's really mostly pushed by the reviewers of my grants. So lots of people that I've worked with over the years, and very fortunate to have mentors that have let me, like this crazy physician that wanted to learn how to pipette after residency, after fellowship, so one of my postdocs, so Chitra Iyer, she did her graduate studies in Arthur Burgess's lab, and so she was a grad student when I made this transition, and now she's a postdoc in my lab, and it's just this really amazing environment to be able to grow in. And so I work with lots of different people that help us develop the virus strategies and the measures, and people that have been mentors in neuromuscular field as well as aging, and I'll take any questions. Go ahead.
I missed the start of your talk, so I don't know if you already addressed this, but is the expression of SMN at all affected by baseline activity in the person? So let's say you don't have SMA.
Yeah.
But you're thinking about aging, you're increasing over time.
Mm-hmm.
You have an extremely active elderly person who's been running marathons for decades versus a person who has a more typical sedentary lifestyle.
Yeah. It's a great question. So actually in the SMA field, they did an exercise study, and they showed that SMN levels go high and also extend survival. We're doing some marathon runner mice studies of motor unit connectivity across the lifespan to look at that question. So I would say the short answer is probably. The problem is it's hard to measure SMN. So depending on where it's going, so it's different in different cells. It's different at different life points of the lifespan. So it's really noisy in patients, and it's not easy to track. So if you look at their blood, it doesn't always even show a difference between types of SMA. So it's not very reliable. So they're working on better measurements of that. But that's a great question, which I think the answer is yes.
So, two questions. What are the main functions of SMA
Mm-hmm.
and are there any evidence that SMA level or gene expression is altered in certain patients?
So the first question is the function of SMN, and that would have been probably really good for me to mention. Sometimes I give talks to people that know this field forward and backwards, so I apologize for that. Second question is whether it's altered in sarcopenic patients or not. So the first question is it's really important for SNRP assembly, so RNA metabolism. So in aging, you do have increased species of RNA, so it is possible that that could be a role. They don't know for sure if SMN levels, when they're low, if disruption of SNRP assembly is the mechanism of SMA. We don't know why low levels of SMN cause SMA. It's a big, huge unknown. It's really amazing. 1,000 different mechanisms have been invoked, but nobody knows. As far as your second question, I think there's two things I would say to that. One, no, people haven't looked at that. The second thing is because we have this differences of genotype in the population, we could actually look at that genetically in humans to look at dosage of SMN1 and SMN2, because SMN1 actually is duplicated in some people. So some people have 3 copies of SMN1. And would they be less susceptible to sarcopenia? So those are questions that I hope to be able to address more in the clinic with databases like there's a Health ABC database of older adults aging, and they have all kinds of phenotypic readouts. But the animal will give the rationale for NIH to let us use that database. So that's down the road, but I hope we can address those questions. Mm-hmm. It's actually my backyard. So this is okay. So my primary goal in the last 6 years has been to become an independent physician scientist. I got my own independent lab space within the last year. So most of this, I was squatting in other people's space, which it's fine. That's actually usually I would say starting out that's the better way to do it. But this year was really an amazing year. But my other big goal, and sometime in my lifespan, 2 years ago I did a relay across the English Channel. But sometime I want to do that solo. So this is Dover, England, and one of my favorite places, really cold water. But I hope to go from there to Calais, France one of these days.